Thank you for joining for today's Coffee Break webinar. Today's topic is Modeling Pediatric Epilepsy with iPSC-based Technologies. Epilepsy is a devastating neurological disease caused by an imbalance in the electrical signals between the cells of the brain. It affects 1% of newborns and very young children. In children, a third of all cases fail to respond to currently available medication. The seizures can result in long-lasting delays in learning, thinking, and emotional development, even early death. Doctors often have to resort to a trial-and-error approach because it is hard to predict how a patient will respond to a particular therapy. This can have devastating implications for young children and their families. The primary challenge in the fight against epilepsy is the lack of model systems that effectively mirror what goes wrong in human patients. Scientists have relied on using non-human cells or animal models of the disease, but fundamental differences exist between humans and animals in the biology of the molecules that are implicated in epilepsy. To solve these problems, Dr. Kiskinis' lab generates patient-specific stem cells and differentiates these into patient-specific brain cells. By studying their electrical patterns and how they respond to drugs, Dr. Kiskinis hopes to be able to predict what drug would work best for each patient. Removing the trial-and-error approach to epilepsy therapies would have a major impact on how we diagnose and treat epileptic patients. Dr. Kiskinis is an assistant professor of neurology at Northwestern University Feinberg School of Medicine. He also serves as the scientific director of the Stem Cell Core Facility and is the co-director of the Stem Cell and Regenerative Biology Initiative. His lab aims to identify points of targeted and effective therapies for epilepsy and ALS by using human pluripotent stem cells.
Thank you for that kind introduction, Melissa. Each year, about 1 in 100 children are diagnosed with a form of pediatric epilepsy. In the most severe cases, babies start seizing within hours or days after they're born. The seizures, which result from aberrant electrical activity in the brain, are multiform, relentless, and exceptionally hard to treat. As a result, these kids suffer serious comorbidities, including cognitive, behavioral, and neurological deficits. Clinicians have no effective way of selecting the right anti-epileptic drug for its individual and often resort to a trial-and-error approach, which can have detrimental effects for babies that are in developmentally very sensitive stages. Pediatric epilepsy, which is what we're interested in, has extensive genetic diversity, with 50% of cases caused by de novo mutations in genes that encode ion channel proteins, which control neuronal excitability. These include sodium channels and potassium channels. In my lab, we've been developing programs looking at a number of these variants using iPSC-based approaches. Now, I believe that iPSC-based technologies are very well suited for the study of pediatric epilepsy for a number of reasons. It is really hard to create animal models that accurately recapitulate the human condition because A, several ion channels exhibit human-specific expression patterns, and B, the clinical severity for these syndromes depends on the interaction of each genetic variant with the patient's-specific genetic background. Now, iPSC-based models retain the genetic background of patients and allow us to examine distinct neuronal subtypes, breaking down a complicated system like the brain in smaller parts. iPSC-differentiated neurons resemble neurons of late embryonic, early postnatal stages, which is the time at which seizures begin for these syndromes. Most importantly, these models allow us to determine the effects of variants on neurodevelopment and excitability, and evaluate the ability of small molecules to reverse or slow down these defects. Now, our goals are to develop biologically relevant and screenable models of epilepsy and use these to stratify patients for optimal drug selection. I will use our work on KCNQ2 associated epilepsy as proof of principle to illustrate the range of scalable assays we've been developing in order to improve drug selection for these kids. Now, the KCNQ2 gene encodes a 6 transmembrane domain protein that is responsible for neuronal M current. There are currently more than 300 distinct variants that have been associated with KCNQ2 associated epilepsy, and clinically, they're all treated in the same way. Now, the first step in our analysis platform is a high-throughput functional assay that combines heterologous expression of either the wild type or the mutant protein with automated wholesale patch instrumentation. This is an example of primary data with the activity of the wild type or a mutant protein. Now, this system allows us to collect measurements from hundreds of cells and screen through a high number of variants in a relatively short amount of time. We use this functional data to classify variants based on their pathogenic effect on the channel. Some are benign, some cause severe loss of function, some cause mild loss of function, and some cause increased activity. We then select representative variants from each one of these groups and analyze their effects in patient neurons using iPSC-based technologies to gain further mechanistic insight on how these variants impact neural development and function. This is the second step in our analysis platform. Here's an example of two patient mutations classified with severe loss of function effects. We generated patient-specific induced pluripotent stem cells and then used these to differentiate them into cortical excitatory glutamatergic neurons. One of the assays that we developed allows us to measure functional M current, i.e. the function of this protein, which is something that had never been done before in human neurons. We do this by wholesale voltage clamp in combination with pharmacological inhibition. We find that while the density of current is consistent amongst neurons derived from 3 unrelated healthy control individuals, it exhibits a robust reduction amounting to 55% in neurons derived from patient 01. Importantly, correcting the mutation using CRISPR-Cas9 restores protein function to a level that is equal to control neurons, while the second patient mutation showed a similar reduction. These findings are really exciting for us as they provide us with a human neuronal platform to screen for effective channel agonists, which have been proposed as therapeutics for these patients. We've also been developing assays to measure the functional outcome of these mutations on neuronal firing. Children with KCNQ2-associated epilepsy exhibit a characteristic pattern in their brain EEGs known as a burst suppression activity pattern. They specifically show highly synchronized firing, referred to as bursting, followed by intermittent periods of very little activity. Now, to assess the firing pattern of iPSC-derived neuronal populations, we use multi-electrode arrays, culturing neurons on fitted electrodes, and recording spontaneous firing over several weeks in culture. We find that patient neurons exhibit a significant and time-dependent increase in the firing rates, with a higher number and percentage of synchronized bursts. In fact, representation of the iPSC-derived neuronal data in a raster plot highlights a burst suppression firing pattern that is highly reminiscent of the one seen in the brain EEG of the same patient, effectively recapitulating epilepsy in a dish and providing us with another phenotypic platform that can be used to assess potential therapeutic options. We have also established an optical physiology assay that enables high-throughput phenotyping of thousands of patient neurons. This is referred to as the optopatch, and it relies on the co-expression of two proteins, a channel rhodopsin that triggers neural activity in response to blue light, and an arch rhodopsin that emits precise amounts of red light in response to neural activity. This movie shows the neurons expressing these constructs firing spontaneously and in response to stimulation. By collecting the red fluorescent signal and applying image analytics tools, we record various excitability features with single-cell resolution. On the right is the recording from approximately 3,000 patient and isogenic control neurons under a blue light step protocol, where we gradually increase the stimulation intensity. At the bottom is the aggregate data which shows that the patient neurons exhibited increased firing frequency under all conditions tested. With this technology, we capture a number of excitability features represented in this radar plot, such as frequency and spike width. The red line represents the average properties of patient neurons, and the blue line, the properties of isogenic control neurons. We can also measure the ability of small molecules to restore these defects. As an example, when we apply a casein Q2 channel agonists, we can rescue a number of defective neuronal features, such as frequency and adaptation, while others that represent dyshomeostatic, likely secondary effects of channel dysfunction, are unaltered. In summary, I've illustrated how we've been developing tools and assays with a goal of impacting pediatric epilepsy research and treatment, a disease that's been largely overlooked and is particularly amenable to modeling with iPSC-based technologies. I believe that this will be invaluable to the community, as the entirety of what we know about these syndromes is based on limited non-human based model systems. Here's where I see this work in 5 years. Firstly, we aim to analyze as many as 1,000 epilepsy-associated genetic variants and classify them based on their effects on function. Secondly, we'll provide mechanistic insight into how at least 50 representative variants impact human neuronal subtypes using a range of approaches. Thirdly, we'll determine whether iPSC models can be used to select patient-specific drugs by performing drug treatment experiments on patient-derived neurons. In collaboration with neurogeneticists and computational biologists, we'll integrate our datasets to stratify pediatric epilepsy cases. Our goal is to enable clinicians to rationally and effectively treat these kids and push through precision care in epilepsy. The work that I presented today was done in collaboration between my lab and Al George's lab in the Department of Pharmacology here at Northwestern University, and was spearheaded by Dr. Dina Simkin and Dr. Tim Searle. We also collaborated with Carlos Vanoy and John Milichup, who provided all the primary patient samples from which all our iPSC cell lines were generated. I also want to mention that all the optopatch-based work is in collaboration with Louis Williams, Graham Dempsey, and Owen McManus at Q State Biosciences. And I also want to thank our funding agencies that have supported this work.
And that is the conclusion for today's Coffee Break webinar. If you have any questions you would like to ask regarding the research presented, or if you are interested in presenting your own research with microelectrode array technology, please forward them to coffeebreak@axionbio.com. For questions submitted for Dr. Kiskinis, he will be in touch with you shortly. Thank you for joining in on today's Coffee Break webinar, and we look forward to seeing you again.